Okay, so let's get started. Well, first of all, probably my congrats. So today we start our last lecture of the course, so lecture 15. Tomorrow we will have the second part of this lecture, and I guess on Friday we will finalize our course with some sort of simple exam, and that will be it. And I guess we really have what to be proud of, because that was really long and maybe in some sense complicated course. At least it was for me, because when we started I didn't have even a single slide or a single problem to give you for solving, and now I guess there are a few hundreds of different slides and materials and almost 100 problems, so that was really a lot of work was done. And of course you have solved a large number of problems and went with me through a whole lot of material, and I guess that's really that's really a thing to be proud of. So I hope you got some new ideas from this course and some new knowledge. Maybe you were able to look at something with some sort of from a different standpoint or something like that, and I hope that you will benefit from this course and from all that we were able to get through. Yes, these all material that was created on the go, so some things maybe a little bit too raw. Of course, I didn't have like timings or something like that, so I had no idea whether I'll be able to fit that material into the time slot we have, or that will be too much or too little. But anyway, we managed to get through all of that, and that's really good. And well, I hope you will really benefit from all that, from all these information we were able to get through. Good. So today's lecture continues this sort of neurobiological part of the course. So as you know, I like neurobiology and neurobiochemistry, and I guess it's a that will be a nice lecture to finish everything, to consider probably the most complicated system that we have in our brain, the system of vision. So how do we analyze the whatever we see in the world around us, and how that helps us to create some sort of mental model of the environment. Vision is really, really important for us. So most information about the environment we get from visual stimuli. So I can't give you some exact percents, and anyway, I think that all these estimations are a bit flaky. But anyway, if you wish, let's say 90% of the information about surrounding environment comes from vision. And what more underlies the importance of vision for us is that more than 50% of the cerebral cortex is involved in visual processing. So you know that brain is really expensive. It's extremely expensive in terms of energy that you use, in terms of oxygen that you use, and so on. So you probably will never ever spend like 50% of that computational power to do something meaningless. So it's really, really important for us. What's interesting, vision is highly contextual. So it's not just getting some image that we obtain on our retina. That's basically the problem of the contextualization of whatever we see. So that image is structured and transformed in some sort of model of the world. And I will show you just, I guess, on the next slide, I will show you some example of that. And I hope you will like that sort of example. So just wait a moment for it. That should be nice. And another take, well, along these lines, is that we rather see with our brain than our eyes. So there is a lot of processing in our brain, and in some sense, brain even does certain predictions of the future. So people are capable of performing certain movements that cannot be based on the image that they see right now. So these movements should be based on some sort of future prediction. So that takes into account all the time that we need for transferring signals and so on and so forth. Moreover, even right now, when you are when you are looking at screen or anywhere, our eyes are moving. Well, more or less, they are moving to the left, to the right, so we have seen something and so on. And the thing is, why don't we see any blurriness? The thing is that there is so-called saccadic masking. So the brain selectively blocks visual processing during eye movements in such a way that neither the motion of the eye and subsequent motion blur of the image nor the gap in visual perception is noticeable to the viewer. So we are moving our eyes, but we do not see any blurry image because our brain has masked this. If you take your phone and try to make a photo or something like that, so if I take my phone, I try to do some photo moving it, I will definitely see a blurry image, and that will be not okay. But you can't do that just moving your eyes. So we have a lot of different processing. Oh, I'm sorry. And here is the example I want to show you. That's example of the contextualization. So whatever we see is in our brain is transformed into some sort of interpretation of the world around us. And here you will see that's some sort of experiment. Whatever you see here is just some sort of cardboard. It has a few holes punched in that cardboard. And here is a ruler, so just a wooden ruler, that is just passing through one of these holes. This thing will rotate. And whatever I want you to look at, whether it will look for you like this ruler and the cardboard rotate in something like opposite directions. And let me know in chat whether you see this sort of effect.
And I'm going to put a ruler right through the middle of the window, and so we can't possibly be fooled by the illusion, right? Okay, here we go.
So here turning starts.
Okay, the ruler is rotating around.
So basically the ruler is attached to this sort of cardboard. And cardboard with a ruler rotates just one direction. But if you look at this sort of video, you might see that they rotate something like opposite directions. Now let me know in the chat, have you seen this sort of effect? Does it look strange for you? Yes. Good. So you're more or less able to see this sort of effect, and that's basically the result of our brain reinterpreting this image in the context of the three-dimensional rule. Yeah, that's absolutely correct. So it looks like they are some sort of, I know, rotating in different directions, and ruler is like pathing through the window and so on. What's more interesting, we can escalate this. So let's make it even more strange. So we were using a ruler. Now let's use a living human. So why not? Human experiments on the go.
So then I would twist up these metal cables that hang it to the ceiling, and then jump in the window and let them unwind. Okay, so I'll show you some of the best shots I was able to get, and you let me know, does this work for you? Do you see it oscillating, or do you just see it rotating, as it actually is? I found lighting is really important. The lighting needs to be really even on both sides to convince you that it is really oscillating.
Well, were you able to see the effect here? Yes, that's basically the result of our brain, like interpreting the three dimensions of our world, and well, let's say we have certain expectations from the environment, like right angles or something like that, and if we use this sort of frame and we color that appropriately, we can trick our brain into thinking that its 3D position is pretty different from what it really is. So this illusion is called Ames window. These are excerpts from the Veritasium channel, so you may want to check that if you wish, but I think I have cut out the most interesting part. There are some different illusions based on 3D. I guess there is Ames rule. Probably yes, it's the same Ames, I guess. That I don't have a video right here, but you can look that on the internet once again. Can we turn off this effect to see normally? I'm not sure. I guess this is one of the effects that you see even being aware of that effect. So I don't think that you can do it. Maybe some long training, but I'm not sure. the We have a pretty similar effect with Ames room when different object sizes look very different from what they really are. So that's probably one of more such illusions. I don't have it right now at hand. Maybe I will show that to you tomorrow if I don't forget. So I write it down. Maybe I'll show you Ames room. But the reason is that we are living in a world of different right angles, so probably this is one of the like reinforced learning on our brain from the surrounding environment. I'm not sure if this effect will be seen with some, I don't know, maybe some indigenous people somewhere in the Amazon River. So if they don't have some sort of civilization with that right angles, maybe they will be much less error prone in sense of seeing such effects, but I don't know. And I'm not sure if anyone knows, so probably we can check some articles somewhere, but but at the moment I have no idea. So so now you see that visual processing is pretty complicated. So that involves not only detecting light, but interpreting that with our brain. Of course, this process is, let's say, not studied up to the end. So we know some parts of it. We know something, but we don't know everything. And at least some parts of this process I want to highlight, of course, in the context of the biochemistry, and maybe show you some known pathways from neurobiology. Okay, so the first station of the light that comes to Earth from the environment, that's of course our eyes. So here you see the skin of the eye, and here you see different parts of the eye. So here you see light, it's coming, it goes through cornea, it gets into the vitreous chamber, it should go through the lens. This lens helps us focusing this light. Interesting part to notice, if you look at the coefficients, these like n, you will see that they are almost equal for the cornea, for the vitreous body, and so on. So lens has almost the same n as the vitreous body. So just some sort of percents. So most of the light bending is performed basically by the vitreous body, and just a small percent of that may be attributed to the lens. So that's maybe some interesting point for you. Here is the retina. Retina is performing some sort of detection of the light, and we will get into some sort of biochemical processes of that detection. You see near the retina is so-called pigment epithelium. You may already know that we have a little bit a strange structure of the retina, where the cells that are basically detecting light are down there under few layers of some different cells, including the ones that are transmitting signals. And the reason may be that these cells that are detecting light, they want to contact with so-called pigment epithelium. This is once again the case when you need to have some sort of very large metabolic rate in certain cells. These are the photoreceptor cells, and they cannot handle that on their own. So they are sort of exporting certain things to the pigment epithelium and getting from that epithelium some chemicals that they need. So once again, this is a case when one type of cells cannot handle the metabolic burden that they carry, and they are trying to share this burden with some nearby cells, and that nearby cells, they perform at least part of the work, part of biochemical synthesis for these cells. So that may be the reason why we have our retina like this, despite it looks pretty strange. Here you see once again this scheme a little bit enlarged. Here you see the photoreceptors. We will get into a little bit more details just in a moment. And here you see different other cells that help to get all these signals from the photoreceptors, a little bit even integrate that information, and then send it to the brain. Two things to notice. The first one is the fovea. This is the place of the sharpest vision of our eye. When we are looking at something and we want to take a good look of that object, we will position our eye so that the image of that object will fall into the fovea. The second thing to notice, you probably know from the biology course, from general biology course, that we have so-called rods and cones in our eye. So rods, they let's say have no color specificity in certain sense, and they are mostly used at some dimmer light, while cones they are used at normal light, and they are specific to certain wavelength, so that we can distinguish colors. So these fovea contains a lot of cones and just a little bit of rods, so a little bit to none. So here we have the best color vision that we may have. But if we think about some dimmer light, is this a good spot to try to look with? How do you think? Probably not, because we have almost no rods there. Yeah, you're absolutely correct. No, that will be a bad place to focus light, because there are almost no rods, so we will not be able to detect anything. That's good. Here is some sort of model of the lens. So I just want to show you this sort of model. I found some nice videos on the internet and once again made an excerpt of that video. So here you see water is pumped inside the lens to mimic that sort of change of the shape. Of course, in the eye we have some sort of muscles around this lens, and we are trying to squish it or to unsquish it. And depending on that, the form of the lens, its shape, it changes. Here it's modeled by just some sort of pumping water inside or pumping it from there to the outside. And here you can see how this how the thing works with some sort of simple object. So here you see an image of the object, pretty blurry, but now he will try to change the shape of the lens, and here you see how that blurriness, well, practically goes away. So at the end of the video, well, I'm not sure why can't I stop it at the end. So what's wrong? Okay. Okay, let it be. I guess you believe me that it was almost sharp at the end of the video. Good. So now returning to the eye. Here you see the light that gets basically to the retina, and here you see different layers of cells in the retina. Note that light should pass a few layers of cells before reaching photoreceptors. So here are these different layers. So you see here the ganglion cell layer, some inner nuclear layer, and so on. And here are the photoreceptors. These are rods and cones. Here I guess we have some sort of micrograph. So this is a micrograph of whatever is happening right there. It's a little bit turned upside down. So here on the top part you see the photoreceptors. These are rods and cones. So here you can see them, and these are the different types of cells that you can find in the retina. So here they are. Once again, kudos to all those guys that are able to read these sort of micrographs, because that was always a sort of problem for me. Well, yeah. Here you see sort of plot that tells us something like number of rods and cones in different parts of the eye. So in the central retina, relatively few photoreceptors feed information to a ganglion cell. In the periphery, many photoreceptors provide that input. So this arrangement, it makes the peripheral retina better at detecting dim light, but the central retina better for high resolution vision. So if you have few photoreceptors passing signal to one cell, there's a higher chance that at least one of them will capture some photon and will generate some sort of signal to the brain. But on the other hand, they are all coupled together, so you don't have so nice resolution as you could have. Moreover, looking at this plot, what you can see, first of all, here is the fovea. That's the place where you have most of the cones. So this micrograph showed exactly this place. So you have many cones here. And almost no rods. So almost no rods. In other places, you don't have so much cones, but you have a lot of rods. So on the periphery, you will see something like this. So this is a typical picture for the periphery. So here you have cones, and here you have different rods. What's more interesting to notice? Here is so-called blind spot. This is a place where all these neural endings go out. So we have some sort of ganglion cells, and they should send their signal to the brain. And here you see this place, this optic nerve comes into the eye, and that's the place where we have no photoreceptors. So basically, that's a blind spot. We cannot see anything with this part of our eye. It may sound a little bit strange, because you may tell me, well, if I close one eye, there is no blind spot. So I'm looking and nothing is missing. But actually it is, because our brain is performing a lot of different processing. And here is an example for you. So let me check it for myself. Yeah. Let's do an experiment. So you close your right eye. You can use a hand, for example, or just close it, whatever makes you comfortable. Then look at the cross at your screen and try to adjust distance to the screen, like moving back and forth, and notice what happens to this sort of circle on the left. So closing your right eye, looking at the cross, and then adjust distance and pay attention what happens to this sort of circle. It's gone. Yes, absolutely correct. It's gone. It disappears. That's basically the distance when the image of the circle gets into that blind spot. Our brain is tricky, so it tries to process the environment and hides from us the existence of that sort of blind spot. But it's there. It's there, and we are able to measure that with this sort of simple experiment. So that's pretty possible. Of course, in the right eye you have the same thing, but it's just symmetric. So if I do this thing in reverse, we can retry experiment with another eye, and that will give us pretty the same result. Good. So now we have seen few different experiments, and we more or less know that there are certain subtleties to the processing image with our brain, and basically with experiencing all that. But let's get through a little bit of more details of the on the molecular level, because I assure you there are certain neat things as well. So as always, I love to position where we are in the organism, and then going down up to the molecular level, when we can basically consider some sort of biochemistry. So now we are positioning ourselves even a little bit deeper. So we were in retina, good, but retina contains a lot of different cells. Okay, now we are interested in pretty particular types of the cells that are basically photoreceptors. And here you see them. Here is a rod, and here is a cone. Here you see in the cytoplasmic space there are some sort of free-floating discs. These are in rods. And here you see some sort of folding of the outer membrane. These are in cones. So the outer segment consists of a stack of membranous discs that contain the light-absorbing photopigments. So the biochemical magic will be performed basically on either on these discs in rods or in these sorts of foldings in cones. So whereas so that's what we basically have. Here we have some even closer look at whatever we will be dealing with. I guess not at this slide, but after we will consider some sort of mechanism of capturing the light, we will get through the signaling cascade that involves different proteins that are basically stationed on these discs. That's for a rod. But you may imagine a pretty similar image for cones. So that's pretty similar. And here you see how that looks in case of dark and light. So if anything is dark, you have these sort of proteins, rhodopsins, they are inactive. Sodium channels are open and the cell is depolarized. Whenever light comes, they react, some sort of signaling cascade goes on. We will consider that in more details in a couple of slides. Sodium channels, they close, and the cell hyperpolarizes. In case of depolarized cell, we have some sort of constant glutamate release. In case of hyperpolarization, this glutamate release is reduced. And that basically reduction in glutamate release signals that we have some photon, and that photon came to our eye and we need to process that somehow. And here is basically the rhodopsin. That's what happens when the light comes. So here you see this molecule that's retinol. I will show you some more details once again in a couple of slides. When the light comes, it changes its conformation, basically rotates around the double bond, using energy from the light. And that changes the conformation of the protein. So here you see the meta-rhodopsin 2. This molecule is covalently connected to the protein. It's connected to the lysine tail, and this is some sort of Schiff's base. I will show you the formula, I guess, and basically in this slide. So here you see the protonated Schiff base. Schiff base because we have this part, so double bond nitrogen. It's protonated, it has this proton, and with this proton comes some positive charge. It's linked to the lysine 296 in opsin by a Schiff base linkage. In the resting state of rhodopsin, this Schiff base is protonated, as you see. Here you see this is the nitrogen, so that's the part of the lysine, so it goes somewhere to the protein and there is a whole large protein around it. Here you see the double bond of interest, and this is 11 cis retinol. So here is 11th carbon and it's cis retinol, so these parts are on one side of the double bond. When the light comes, it transforms into all trans retinol. So here you see the change in this molecule, so how it transformed from the 11 cis retinal to the O trans retinal, and this sort of change will be able to be transferred to the protein, and then some sort of cascade goes on and on. I will show you the details just in a moment. But before that, what's interesting with this sort of retinal? If you look at these chemical formula, you can see a lot of double bonds that are interleaved with single bonds. So you see a single bond, a double bond, single bond, double bond, and so on. What's interesting in this sort of construction, you will see this sort of chemical feature in many natural pigments. So here you see the lycopene, that's a red pigment of tomatoes. So do you see this pattern? Double bond, double, single, and so on and so forth. Here you see lutein, a yellow pigment in egg yolks. Once again, double, single, so the same pattern. Beta carotene, so an orange pigment in carrots, and this is the chemical pretty similar to the to this cis retinal. So basically, two cis retinals can be made from beta carotene. From one beta carotene, by some sort of like removing one of the bonds. So where is it all basically going? Here is once again a place to remind you about whatever we started with, about basically the chemistry, the orbitals, and so on. So I hope you remember this sort of thing. So we started with some sort of quantum mechanics. I told you that when we have different atoms and they combine into molecules, they obtain something like molecular orbitals. So from the atomic orbitals, we are going to molecular orbitals. These molecular orbitals were computed, well, let's say approximated with us with some sort of linear theory. So we were like linearly combining different wave functions for atomic orbitals to get something new, to get some molecular orbital. And you remember that combining wave functions, so wave function is wave, so you can combine that constructively and get some larger wave, or you can combine that destructively and get basically nothing. And here is an example of combining something like two hydrogen atoms, where we get two new molecular orbitals. And here is a constructive interference, so we have something like this, and this orbital will have some lower energy than the atomic orbitals we started with, and some sort of destructive interference called anti-bonding molecular orbital. And here you see its form, and it will have some higher energy than the orbitals we started with. Since we are populating just with two electrons, these two electrons can live on the lower constructive orbital, on the bonding molecular orbital, so we have some gain in energy, and these two hydrogen atoms, they would like to stay together, to combine together into a molecule in H2. And this is the case for helium. This case is a bit different, because once again we have bonding and anti-bonding orbitals, but now each atom is carrying two electrons, so these electrons will use up the bonding orbital. Due to the Pauli's exclusion principle, we will need to put another two electrons that we have into the anti-bonding orbital, and basically we have no gain in the energy. So helium does not like to combine into molecules. So that's why you will always see if hydrogen gas, that's H2, if helium, that's just helium. It's not helium-2 or something like that. So that's the reason. One more reminder, that's once again we are getting to combining all these orbitals and so on. We may have so-called sigma bonds and pi bonds. So I'm just reminding you of whatever we have learned already. So here you see a double bond between two carbon atoms. So one bond is sigma bond, so here it is. The other one is the pi bond. Here you see this pi bond in the terms of energy, and once again we have bonding and anti-bonding orbitals. One is pi, one is pi star, so whatever is anti-bonding, that has a star. This is in case of different atoms. So where is this all going to? Well, it's going to the conjugation. So here we have a butadiene. So this is that sort of pattern. So double bond, single bond, double bond. Well, and that's it, because I tried to use the smallest molecule that has this sort of pattern. And here you see what happens. These double bonds, that's basically what? That's one sigma bond that is connecting two carbons. And here you have the two perpendicular orbitals that are basically pi bonds. And in this part, you have the same thing. So I have these sort of pi orbitals sticking in the perpendicular to the plane of the molecule for this double bond and for this double. So of course you might think, why don't these two interact? And electrons think the same, and they say, well, there's almost no difference, why don't we interact? And they interact. And that leads to pretty interesting consequences, like changes in different energies of these sort of molecular orbitals. So here you see them on the right. So here are different energies when we are combining everything in phase, when we are combining something out of phase, and so on. And we are populating these molecular orbitals with electrons. Now, where it gets us to? It gets us to the question of the energy between HOMO and LUMO. Who remembers what is HOMO and what is LUMO? Yep. So this is highest occupied molecular orbital. This is lowest unoccupied molecular orbital. When a photon comes, it takes electron from the highest occupied molecular orbital and transports that to lowest unoccupied molecular orbital. So this way, molecule may absorb a photon. When we are, let's say, engineering some sort of biological photoreceptor, that's what we want. We want some molecule that will absorb a photon and something will change. Then these changes will be somehow detected by our system. So this promotion from HOMO to LUMO, that's basically absorption of the electron. And what I want you to show here, so here is ethene, it has just one double bond, and here we have some gap between HOMO and LUMO. To carry an electron through this gap, we need some photon in a far UV spectrum, something like 185 nanometer. So that's far UV. So here is the scale on the right, and here you see the ultraviolet. Here is the visible light, so all the colors we know, and then it's some infrared below, and so on. When we add this sort of conjugation, we are decreasing the gap between HOMO and LUMO. So now, just getting from ethene to butadiene, we are able to change this gap from 185 nanometer to 215 nanometer. So I hope you remember, so the longer the wavelength of the light, the lower is the energy. So now we can have this transfer of the electron just with 215 nanometer. And we can go even further. So here is a spectrum, and here you see this sort of structure like when we have these conjugated bonds. And here you see the color that is basically absorbed. So you can absorb some violet, indigo blue, blue, and so on, depending on the number of that conjugated bonds. So that's the case. So what the idea? This molecule is like so because we want a lot of conjugated bonds to make small HOMO-LUMO gap to be able to use visible light to promote electrons from one molecular orbital to another molecular orbital, and that will give us the ability to change this molecule from that cis to trans state, and then propagate this sort of signal through through a signaling cascade that will eventually lead to transferring information into the brain. So what I want you to notice, we are now basically recalling whatever we have already studied. So you don't need to think like we have studied some sort of chemistry and that's it, and it's gone, so we studied, passed some tests and forget about it. No, it's still, everything is still here. It's still working on a little bit maybe different level. So all that quantum mechanics I told you about, all that conjugated bonds, all that sort of different chemistry, it's still here. It's still working, and it leads to this sort of complex behavior and these sort of interesting changes in the molecules. So whatever we are looking here, it's some sort of part of quantum mechanics, because we want some small gap, small enough so that we can use photons of visible light to be detected. Here is the rhodopsin. So it's highly sensitive to light. Its quantum efficiency is something like 67%, meaning that for every 3 photons absorbed by rhodopsin, 2 of them will result in isomerization of retinol from the 11-cis to O-trans conformation. Here is the number of the model. That's from PDB database. If you wish, you can check it there and rotate in 3D and so on. Here's once again the formula reiterated when 11-cis retinol is transformed into O-trans retinol. Good. Now taking a more closer look. The question is, I guess, let me check it, yeah. Now a very, very interesting question. The retinol is the same in rods and in different cones, but they have a pretty different absorption spectrum. So it begs the question, why the same molecule, the same light? What's different? And the answer is that different are opsins. These are the proteins that that retinol is connected to. So here they are. Here you see different absorption spectra for rods and for 3 type of cones. Some people call them RGB, like red, green, blue, which is basically the reason why we have red, green, and blue in our screens. So that's interesting to point out. Or some call them SML, like short wave, these are blue, medium, these are green, and long wave, these are red. So here they are. Color vision is pretty interesting. Most mammals, they see only 2 colors, most of them. Humans and certain primates see three colors. It's probably the reason is that we were for some time looking for fruits, and it's pretty, let's say, important to see different colors so you can distinguish different fruits and not eat something inappropriate. But we're definitely not champions. So probably the champion is mantis shrimp, that has 16 different types of cones and is able to perceive, well, a lot of colors. Each of the four visual opsins binds the same chromophore, 11 cis retinal, as a protonate cis base, so the same structure. And all opsins are 7 helix transmembrane proteins and are members of the G protein coupled receptors. So GPCRs we have already considered, and this is the example of the GPCRs. Here you see once again enlarged view of a rhodopsin that basically consists of opsin, a protein, and a small molecule that is derived from the vitamin A. So vitamin A is needed for us, well, to synthesize that cis 11 cis retinal. Here you see some sort of evolution of different rhodopsins. These are comparisons of proteins that we have in rods and in cones, or let's say opsins. So here you see rhodopsin, that's opsin found in rods, and M means M opsin, that medium wave opsin, or the one that is responsible for the green light. They are compared here, and here you see different amino acids, and they are colored depending on the fact do they coincide or not. So in case they do not coincide, they are blue. Here you see comparison M versus S, that's medium wavelengths or green versus short wavelengths or blue, and here you see L versus M. A lot of difference, but actually they seem to be pretty similar in the 3D structure. Okay, or I'm missing, and oh no, I'm sorry. I guess I'm missing, and blue is actually total coincidence, while not blue is difference. So I guess, yeah. And L and M, they are very closely related, like with 96% of the identity in their amino acid sequences, and they are thought to derive from a gene duplication event approximately 30 million years ago. So after the old world monkeys, which have 3 pigments, separated from the new world monkeys, which generally have only 2. So that's the case. I guess you should have studied some sort of mechanisms for all that duplication during, I don't know, maybe molecular biology course or genetics course, I'm not sure. Okay, so we have different options, we have different proteins, but still the molecule that absorbs light is the same. So what is the mechanism to make it absorb like different wavelengths? And I'm not sure if at this point anyone knows exactly, but the last time I checked, there were at least 3 models. Well, one of them was thought that it's probably not the case, but another 2, they look like pretty probable. They are illustrated in here, so A, B, and C. These are different mechanisms to make it absorb different wavelength, and the A part, that's basically the model that says that rotation along single bond C6, C7. So here it is. So this one. Or twisting along double bond C11, C12. So here it is. You see these red arrows, and they basically show you where the rotation is performed, could alter the degree of the conjugation. To be conjugated to delocalize the charge along the molecule, you need to keep this molecule planar. So you want all the carbon atoms to live in a plane. If you somehow twist the parts of the molecule, and you can do it because you have the protein all over the place, and probably different parts of the protein, they may just create some sort of steric hindrance and somehow distort this perfect planar shape of the molecule, and that will change the degree of the conjugation, and that will of course influence different energy levels that we have in this molecule, which of course influences what is the wavelength of the light it should absorb to transform from 11 cis to all trans. So that's one model. Another model we have that's a resonance of the positive charge along the polyene provides the opportunity for interactions at different locations to stabilize or extend the delocalization of the positive charge. It has been hypothesized that different opsins could alter the wavelengths of absorption through electrostatic interaction with the polyene at different locations. So in simple words, you have some charge, some, let's say, negative charge. So look at this. We have a Schiff's base. Schiff's base has a positive charge on the nitrogen. We have a lot of conjugations happening all over the molecule. So these positive charge will be somehow delocalized along this molecule. That influences energy levels and the wavelengths. Now we have a protein. This protein consists of many amino acids. These amino acids, they have some sort of residues. Residues may be charged. So with a charge residue, if you put some charge nearby this molecule, you will disturb the delocalization of this positive charge, and as a result, you will shift somehow the energy levels. And shifting energy levels, you will get different size of that gap between HOMO and LUMO, and of course different wavelengths to be captured with this sort of molecule. The last mechanism is thought to be not very probable. That's basically two different rotameric conformations of the molecule. So here you see basically what changed. So you see this part like this, and here you see it like rotated. It's not very probable, but once again, this may change basically the conjugation in the molecule. Okay. Now, what else I want to show you with this sort of proteins, just some sort of really briefly. Here is different examples of where do we have them encoded. Most cases we have the S opsin, that's the one responsible for the blue light, encoded in some sort of autosome. I will show you more details about the human autosome in the next slide. Another two, like L and M, are mostly encoded in the X chromosome. So here is the case for trichromates, like humans, different apes, and so on. Here are some more cases that they may have like no S, for example. So there are some nocturnal primates that are monochromates and they have no S, and only some sort of one option, something between M and L, and so on. So that's what we have. Here is an example. Here is a pretty interesting example. If you have a monkey that is trichromate, but it contains only one of the genes on the X chromosome. It may happen that, well, first of all the males in the population, they will be, let's say, color blind, so they have only one X chromosome, so they don't stand a chance for having three different proteins. One is on the autosome, so they do have it. The other one is in the X chromosome, they may have it, but the third one, no chance. Females have two X chromosomes, so they do have a chance to have three different options. Some of them, of course, realize this chance. Here is a trichromatic female, and this is basically how she sees the world. Other are not so lucky, and they don't have two different options in their two X chromosomes, so they don't realize this possibility, and here is how they see the world. So just with two types of different options. So here you see the example. Sometimes it is speculated that this may be pretty important for their community, as the monkeys that are able to see all three colors, they are much better at searching for different fruits. And here you see, let's say, this image. So here you see some ripe fruits that are red, and on the left it's hard to say whether there are any fruits, and definitely no chance to say whether they are ripe or not. So so others tend to follow them when it gets to searching for some food. So that may be pretty interesting to notice. Here is example of the here's basically what's going on with humans. Pretty interesting case. So here is the 7th chromosome. It contains gene OPN1SW. That's for short wavelength, and that corresponds to basically blue color. So here you see some sort of image from the internet. So here is this gene. You can find it on the 7th chromosome. So here you see chromosome 7, and here you can even see some numbers that more or less tell you where it is. Here are some exons, introns, and everything we would like to see. Rhodopsin is encoded in the 3rd chromosome. So here it is. Here, once again, you see these sort of details. While X chromosome contains, pretty interesting thing, contains gene OPN1LW, so one gene for rhodopsin. These genes like TEX28, they have nothing to do with vision, and I guess they are not expressed in... these are, I guess, text means something like testicle expressed or something like that, so they definitely not work in these circumstances, but they are there in the chromosome. And then people may have few copies of the green opsin or gene OPN1MW. Despite functional are most cases just first two genes, but anyway, so that's what we have. And what is interesting here, the cell chooses what to be, so to be a rod, to be a cone, and so on. When we start developing our retina, first of all, there is some signal like NRL that leads to developing as a rod, so that influences the expression of that gene on the 3rd chromosome. If there is no NRL, that will be a cone. Depending on the thyroid hormone, low THRB leads to the activation of genes from the 7th chromosome and expression of gene OPN1SW. So you might guess that each cell, it carries only one type of the receptors, and here is basically the table that shows you how do they choose what to express. Well, this is pretty simple. So if we have high THRB, so we get to the pathway like L and M. That's okay, that's not so interesting. Yeah, some signal comes from the environment and basically tells you be a road, be a cone, be whatever. And here comes the pretty interesting part. Here they choose to be L or M. And here is a pretty classical mechanism that maybe you have seen in the molecular biology course, but I will still remind you of that. These are two genes, so one L gene and one M gene on the X chromosome. And how do we choose which one to express? And here are promoters of these genes and the part called LCR. And this LCR part randomly binds to one of the promoters, either to the red one, either to the green one. And only that gene will be expressed where this LCR binds. After the binding, there is some methylation performed, some changes in histones are done, so it's basically it will be made permanent. But anyway, at certain initial point, this attaching is performed randomly, and that basically decides at random whether it will be a red cone or a green cone. It turns out it is much more probable to connect to something that is close, so the probability of getting red genes activated is much higher than the probability of getting green genes activated. Maybe I have here I have a picture that shows you all that RGB, so you might guess what are the probability of becoming red, green or blue. That's on the right, that's part of retina. And here is an example of the CCD matrix in photo cameras. You see that the balance between red, green and blue is different here, so they are not like 1/3 of everything. I don't remember, it's called some filter, I don't remember the guy's name, but that's some sort of filter that is placed over the CCD matrix to filter the color so that appropriate parts of the matrix react only to that color. And here is our RGB in our retina. And probably one more thing to notice, since these genes they are so similar, it often leads to some problems when it gets to the recombination. So I told you that we have the process of the recombination. Moreover, this is a normal part of the mitosis, so we cannot perform mitosis if we don't have that sort of mechanism. And we have even considered that sort of Colliday structure, so I hope you remember that Colliday structure and so on and so forth. But here is an example when things can go wrong. Here you see this is the normal case. So consider just one X chromosome. You might guess that men will be much more affected with the color blindness than women, because we have only one X chromosome, so if something goes wrong there, there is no other X chromosome to substitute, so no chance to fix anything. And here is example of the X chromosome of a normal male, so it may have one L pigment and one M pigment, or one L and 2M, or one L and 3M, so these are all okay, so any of these things are pretty good. But what happens if a recombination goes wrong? Here you see an example like these genes are recombinating, so these part it gets attached to here, to this point, while this part and everything what goes there gets attached to this point. It may happen that we are losing the green opsin totally. So there is red opsin and no green opsin, so it's just gone. Or recombination may create one red opsin and two green opsins. That will work. But anyway, these thing that's a problem already. Or since R and G opsins are so similar, so on that slide you saw that they are 96% similar, there is certain chance that they both will break and you will get something non-functional, like half green, half red, and who knows how that will work. So these are some sort of pathological cases when basically this gene is broken and something strange is expressed or nothing is expressed, so something gone wrong. So basically this is the main reason of the color blindness. So the process of this sort of rearrangement of that homologous recombination may lead to some problems with genes, like breaking them totally, and this is the case for color blindness. Good. Here is one more reminder. So what happens to the rhodopsin when the light comes? So here you have the opsin protein. Here is retinal inactive. Light came. It changed from 11 cis to all trans, and here it is active. I already told you that this is some sort of G protein coupled receptor. So we have considered many different GPCRs, and we have considered some sort of mechanism, some signaling cascades. So let's do some sort of small comparison. So let's say we have some GPCR and we have some, I don't know, some neurotransmitter that is interacting with these GPCR, and we have, for comparison, we have our rhodopsin and light interacting with that rhodopsin. And here you see a pretty similar case, like we have some G proteins and we have some effector enzyme and we have some second messengers. They work pretty similar. So in case of neurotransmitter receptor, we have some change in protein conformation. It binds GTP, so you know that that G protein that is coupled to the receptor, it exchanges on activation GDP for GTP. Then alpha part goes away. It may activate some different different enzymes. In case of in case of neurotransmitter, we have some increase in second messenger, which then leads to certain ion channel response. In case of rhodopsin, we have decrease in second messenger, and that leads to closure of the sodium channels and decrease in sodium conductance. Here is another example. So here I am comparing common features of signaling systems that detect like hormones, light, smells, taste, so whatever we have studied with. So here is example of the vasopressin. So here we have some receptor, here we have G protein, GI, here is adenylyl cyclase, this is I inhibitory, so it leads to the decrease in the number of the cyclic AMP, that is second messenger, which basically leads to the decreased activity of the protein kinase A. Here you have epinephrine, it's connected to the GS, looks similar, but CAMP goes up, protein kinase A activates. Here is light. Here you see PDE that leads to basically decrease in cyclic GMP that inactivates some sodium channels, so channels they close. Here you have example of the odorant, and here you have example of a sweet tastant. So that's what we have. here is even a closer look what happens at that discs. So I have shown you that sort of small discs, and here is some closer, some molecular example what is happening there. So in the dark, cGMP gates a sodium channel, causing the inward sodium current and depolarization of the cell. So sodium comes in, the cell depolarizes and generates some sort of glutamate, glutamatergic signal, let's say. And this is inactive G protein. The activation of rhodopsin by light energy causes the G protein, it's called transducin in this case, to exchange guanosine diphosphate for guanosine triphosphate, which in turn activates the enzyme phosphodiesterase. So here is PDE, phosphodiesterase, and PDE breaks down cyclic GMP and shuts off this sort of sodium current. Here is even more detailed view of the process of what is happening in there. So that's some sort of molecular view. Let me just check for a moment the material I have here, because is this a good place? Okay. I guess we will stop at the 43rd slide. Yeah, I will show you these slides in details. We do one more small experiment, and that will be a really good place to stop. Good. So let's check just some sort of details of this signaling cascade. Well, we're doing like biochemistry, so we need to get into some molecular details. So here you have the, let's say, step number 1. So light absorption converts 11-cis-retinol to all-trans-retinol and activates the rhodopsin. So activated rhodopsin, well, it's pretty typical for G protein coupled receptors. They do what? They catalyze the replacement of GDP by GTP in transducin. That's a G protein in these sort of rods and cones, which then dissociates. So you remember it consists of 3 parts, alpha, beta, and gamma. The probably most interesting for us here is the alpha part, and the T alpha GTP dissociate from T beta gamma. Then this T alpha GTP activates the cyclic GMP phosphodiesterase. So here is the phosphodiesterase, PDE. It's connected to the disc membrane as well as all these things. And basically it binds and removes its inhibitory subunit. So here it is. So this is inhibitory subunit that activates the phosphodiesterase, and it starts transforming cyclic GMP into this sort of 5' GMP. That reduces the level of the GMP in the cell. So this GMP was basically used to keep sodium channels open. When its concentration decreases, it starts leaving these channels, and channels start closing. So cation channels, they close. That prevents the influx of sodium and calcium, and membrane starts being hyperpolarized, and the signal basically will be passed to the brain. So that's what we have. That's the part of activating the cascade. Interesting point to notice. Everything here is performed on a disk, but nothing is pumped into the disk or out of the disk or something like that. They are just attached to the membrane. They don't need that space inside. The reason for this attachment, I'm not sure if I remember the name correctly, but there is some so-called Poya theorem, and it has to do with random processes in spaces of different dimension. If you have a random process, some random walk on a plane, with the probability 1, you will return to the same point. If you have a random process in 3D space, there is 0 probability you will return to the same point, some sort of random walk. And these proteins, they are moving on that membrane, they are moving randomly. So the probability of two proteins collide and perform something is incredibly higher when they are attached to anything planar. So if you have something two-dimensional, that increases the probability of proteins colliding and performing some actions. So you don't even need some sort of membrane to enclose something meaningful, you just need some sort of membrane to attach their proteins so that they move around that membrane and they have some high probability of bumping into each other and performing something biochemically interesting. So here is that interesting part. Good. Here is just one tricky question for you. It's sort of reminder of our previous lectures. How do you think YG proteins change when activating? Change GDP to GTP. Why not the other way around? It turns out to be much simpler if you just hydrolyze the GTP to GDP and turn on. So why not? Why doing it the other way around? What may be the reason? Do you remember from our homeworks? Yes, that's for the inactivation. It has some sort of hydrolytic activity. If you remember all that structure of the G protein I showed you, it has some hydrolytic activity, and this GTP, after a certain amount of time, will be hydrolyzed and the protein turns off automatically. So you don't have this sort of biochemical cascade ongoing forever, because we never want some cascades to be ongoing forever, because they will be like useless. So you need a way to turn it on, but you need a way to shut it down. So there should be a way to turn it off. So yes, so the hydrolysis of the GTP and transforming that to GDP will automatically turn off the G protein, so that we don't have this sort of infinite signal transmission, and that's really good. Okay, and with that we get to the question, how do we turn off everything? So let's get through that. So here we have some sort of continued efflux of the calcium through the sodium-calcium exchanger. When we turn off all these channels, some time will pass, and the reduction of the calcium in the cell, and it will be reduced because these channels, they were able not only to let sodium in, but calcium as well. Now we have no way to let calcium in, but we have some pumps that constantly remove it, that move it out. So the level of calcium, the concentration of calcium drops down. This reduction activates the guanylyl cyclase. So here is guanylyl cyclase. You might guess by its name what it does. So guanylyl cyclase, it takes guanidine and transforms that into the cyclic GMP. So takes and the concentration of cyclic GMP starts rising, so it rises towards dark level, reopening cation channels and returning the membrane potential to its pre-stimulus level. So it will generate some cyclic GMP. This cyclic GMP will reactivate once again these sort of sodium-calcium channels that will reopen, and everything will return to the normal state. Of course, we should do something with the rhodopsin. The rhodopsin kinase phosphorylates the bleached rhodopsin, so whenever it changes its conformation, it is now possible to phosphorylate it, thus shutting it off. First of all, erestin binds to phosphorylated carbonyl terminus and basically inactivate that. So here we have rhodopsin kinase adds few phosphate groups. Now erestin is bound to the rhodopsin and stops transmission of the signal. Otherwise, we will be constantly activating some new G proteins, and that will be useless, that we were trying to shut something down. So we need to turn off everything, and rhodopsin as well. And slowly erestin dissociates and rhodopsin is dephosphorylated, but the thing is that there are certain steps that remove the all-trans retinol from here and attach 11-cis retinol. And some of these steps of converting all that retinol, so on and so forth, they are performed in different cells, not to put too large burden on these sort of photoreceptors. So that sort of epithelium that we had just below these sort of photoreceptors will be used basically to transport some of these chemicals into there, and they will be processed in that cells. So I will not get into that sort of details because I think it's enough. We have considered this sort of cascade. The only thing I want to show you at the end of our lecture, let's do one more small experiment. You see that we are basically during all these steps we are using up some of the chemicals. So what happens if we have used up that chemicals, and then, well, can we detect that somehow? So now I will show you an image. So this is the image. Contains cross and few different dots. So some of them are white, so they will be like retinol consuming. The other are blacks that are not. And take a look, I guess maybe 20 seconds. It should be a minute, but let's get to 20 seconds. I'll find some clock in here. You should look at the cross, and then I will turn another image, and you should see some sort of after image of these dots on that other slide. So let me find where do I have I have some clock here. So I will measure, let's say 20 or 30 seconds, and then I will switch the slide, and you should see some sort of after image. Good, I hope you're ready, so let's go. Look at the cross. I'm measuring some time, so try looking more or less steady at that cross, and after, I guess, 30-second passes, I will switch the slide, and you should be able to see some sort of after image after looking at all of these things. Ready? Set, go. So now looking at these cross, you should be able to see some sort of after image of the dots on the field. Well, after some time passes, that will basically go away, but I hope you were able to see the effect. Okay, was anyone able to see that sort of after image? Yes. So at least some of you were able to see that sort of effect. So that's because we are using up some of that retinol, so when we are basically switching to another image, we have this sort of after image. So some parts of our retina, they have used the chemicals. Good. Still see dots. That will go away. That will go away just after, I don't know, some small amount of time. Good. There is very similar thing with colors, but I have left it for you for the homework. Yes. Good. So the last slide for today. I guess it's time to wrap up. So what do we have? Some sort of just statistics like something like 50 rhodopsins or 203 cone opsins have to be activated to fire up the neurons and lead to vision. So you shouldn't think that it's like just one photon. No, it's not working that way. Rhodopsins, you guess it's rod opsin, so these opsins in rods, and these are cone opsins. I'm not sure why the language works this strange way. Well, when we get from rhodopsins we get one word, and for cone opsins we have something like two words, but I'm not a philologist, so who knows. The entire process takes only a few milliseconds, so it's pretty quickly processed despite all that chemistry beyond the things. So all the transretinal dissociates and converts to the 11th retinal in the retinal pigment epithelium cells, which are adjacent to the photoreceptors. So that's the part to remove some sort of metabolic burden from that detector cells, because they have a lot of work to do anyway. So this is a pretty common pattern in the biochemistry. The same thing goes to neurons and glial cells when you have some exchange of metabolites, because one of the cells has to do a lot of work and it just cannot operate on its own, so it needs some sort of support in its metabolism. So here you see that retinal cells have a lot of work to do, so they need some support from the pigment epithelium cells. And newly generated 11th retinal binds to apoopsins and reconstitute the 11th retinal bound opsins. So they are exchanged. And that's basically finishes the cascade of the detecting of a photon. Good. I guess with that being said, we are good for today. So if you have any questions or comments, please fire away. Okay, I see no questions, no comments. Good. Then have a nice evening and see you tomorrow.